Hi there, welcome to Phlebotomy Module 1, Lecture 4. In this lecture, we're going to get down to it. We're going to start talking about equipment. We're going to talk about basic equipment for routine collections. We're going to talk about words like hemostasis. That's spelled wrong. It should be H-E-M-O-S-T-A-S-I-S, and coagulation. You'll learn what those are. We'll talk about the difference between, that should be arterial. I need to correct my slides. I will do that. For now, it should be arterial, A-R-T-E-R-I-A-L, capillary, and venous samples. And you'll learn to explain the difference between serum and plasma. So here are your key terms. I'm not going to read through each one of them, but you need to be able to define them for me. So when we talk about hemostasis, what does that mean? That means when there is leakage of blood, a clot needs to form. And then the clot needs to dissolve. Otherwise, your body would be full of clots. So we form a blood clot to stop the blood when the blood vessel is injured, and then the clot is dissolved after the injury has repaired. It happens really quickly. We don't usually think about it. And it's a very complex process. It includes what happens with the blood vessels. It includes little things in your blood called platelets. It includes proteins in your blood called coagulation factors. All of those work together to maintain hemostasis. Without hemostasis, you would be constantly leaking blood. So it's probably good we have this. There's 4 stages in the coagulation process: primary hemostasis, secondary hemostasis, clot stabilization, and fibrinolysis. These are the basic stages. So stage 1 is primary coagulation or primary hemostasis. All that means is that when a blood vessel is injured, whether it's the capillaries in the skin or the veins down deeper or the arteries or an organ or other parts of your body, the platelets rush to the site. Platelets are they're not really cells in your blood, they're parts of cells that their job is to start forming a plug. So they're kind of like the first responders in the coagulation system. So they go right where the body is injured and they form a plug. Then stage 2 is secondary hemostasis. And that's where the proteins in your body that are made in your liver all come together with enzymes and calcium, and they make more of what's called a fibrin clot. And that fibrin clot strengthens that platelet plug and actually makes a good solid clot to keep that injury from bleeding. Then the clot kind of pulls together and waits for the injury to heal. It brings the wound together and helps stop the bleeding. And that fibrin is all cross-linked in there like threads to kind of keep it all knitted together. After the injury is healed, then the fibrin clot gets broken apart. So the clots are dissolved and the injury is healed. Now we're going to talk about sample types. So know that once you are collecting a blood, whether you puncture the skin or injure the blood vessel, the coagulation process starts right away. The body sees that as an injury, the platelets rush to the site, and a clot starts to form. So there's a video attached that talks about the difference between arterial, not atrial, arterial. Hold on a second, I'm just correcting my slide so that I can fix it later. On the difference between arterial and venous samples and you can see the more detailed description on the 2 different samples. So you should know the difference between arterial and venous samples. So a venous sample is the most common. Another sample type is a capillary sample, where it's taken from your fingertips or, in the case of a baby, a heel stick. And then arterial sample. That's right from the arteries. Remember, arteries have a lot of pressure in them because they come from the heart out to the rest of the body. And most phlebotomists don't collect arterial samples. It's usually assigned to a trained respiratory therapist, a medical laboratory science, or a medical laboratory technician. And hospitals will give you further training before you collect those samples. We usually use this to see how much oxygen is in a person's blood or look at the other blood gases like carbon dioxide, and it requires additional training. So you probably won't, as a phlebotomist, be collecting blood gases or arterial samples. So most commonly you'll be asked to collect either a venous or a capillary sample. The venous sample, now remember, the veins take the blood back to your heart after the oxygen is out of them. Out of the blood, I'm sorry. And there's not as much pressure in a vein as in an artery. So the punctures heal quicker, and they're usually closer to the surface, so they're easier to collect blood from. They can be challenging to collect on pediatric or child patients. And then most patients, except small children and babies, we do use a venous sample. For small children and babies or samples that are difficult to get, we get capillary samples. Capillary samples are a mixture between arterial and venous blood. And we're able to control bleeding after we puncture it, but just light pressure. Probably almost everyone has had their finger poked at some point in time. Again, we use these capillary samples when we need small volumes of blood. They're time consuming to collect. You will learn how to collect capillary and venous samples in this course. Capillary samples are ideal for pediatric patients or babies. It's sometimes easier to get a finger stick sample out of a small child than a needle sample. It's less traumatic for the patient, it's less traumatic for the phlebotomist, and it's less traumatic for the parents. Depends on how much blood you need, but most of the time, you'll be getting capillary samples for small children. Okay, so after you get the blood sample, it's classified as one of three types of samples. So it's either capillary or venous, okay? In any case, those are both blood samples, but we may need it for whole blood testing, we may need it for serum testing, and we may need it for plasma testing. Whole blood just means that it is the whole blood, the liquid part and the cell part. So in addition to the liquid part, blood contains red cells called erythrocytes, white blood cells called leukocytes, and those little platelets all suspended in that plasma, just like it is in the veins. We add anticoagulants to keep the blood from clotting because we have to test it, and if it has a clot in it, we can't count those cells. So that's how we get a whole blood sample. A whole blood sample is anticoagulated blood. It's collected in an anticoagulant tube, and it does not clot. So for serum then, there's no anticoagulant in the collection tubes. It's drawn into a tube. Serum is the liquid portion of the blood which remains after the clotting process has occurred. So serum has no more clotting factors in it because they're all formed in the clot, and we'll show you examples of these. You have to centrifuge this tube to separate the serum from the clotted cells, and then we use the serum to run tests. The serum does not contain fibrinogen because that is used up in the clotting process. Plasma, there's anticoagulant present in the collection tubes, and it's a liquid portion of the blood after the tube is centrifuged. So when there's anticoagulant in there, the cells centrifuge to the bottom, and the top, there is no clot. They're still suspended in liquid, and then the plasma is at the top, and we use a plasma for testing. So the main difference is between serum and plasma. Plasma contains fibrinogen, serum does not. We will talk about this more in our laboratory sessions. This is a key concept that you need to know for the rest of your career as a phlebotomist or an MLT. So make sure you understand that definition. Serum does not contain fibrinogen. Plasma does contain fibrinogen. Serum is collected in tubes with no anticoagulant. Plasma is collected in tubes with anticoagulant. So again, this illustrates it. You can see in the serum tube, there's a chunky clot in there. So the serum does not have fibrinogen in it. In the plasma tube, the red blood cells are at the bottom. There's a buffy coat or a thin white line that contains the white blood cells and platelets, and the plasma is in the top, and it does contain fibrinogen. If you turn that plasma tube over, it will remix into whole blood. If you turn that serum tube over, there will still be a clot and it won't remix as nicely. So once blood samples have been drawn and mixed, in order to get the serum or plasma, we have to centrifuge and separate the blood. So in whole blood tubes, we don't centrifuge. In serum and plasma samples, we do centrifuge. So there's a video in this module that talks about processing and aliquoting or separating samples. Be sure to watch that. So in this module, we talked about the basic function of hemostasis and coagulation. We talked a little bit about equipment and types of samples. We'll talk more about equipment in the next lecture. We talked about the difference between arterial, capillary, and venous samples, and we explained the differences between serum and plasma. Again, I'd like to state, if you have questions over anything in this lecture, please bring your questions to the laboratory sessions, and we will discuss it in further detail. Once we start collecting samples and you can actually see this, in addition to the videos, the more you practice, the more you work with this, the more sense this will make. But for now, just understand the key concepts. Thank you, and please review Lecture 5.